Assalamu alaikum everyone. Today we are going to discuss about blood culture, a key tool for diagnosing infections. A blood culture is a laboratory test which is used to detect the presence of harmful microorganisms like bacteria, fungi in your blood. Blood culturing is important to identify the cause of infections like sepsis to guide effective treatment to prevent serious complications to save lives. Step A is collecting a blood sample which require proper steps to ensure safety, and step 1 is prepare the material which requires syringe, gloves, collection bottles. And second step is to identify the patient and explain procedure to them. And third one is to wash hand and wear gloves to maintain hygiene. And fourth step is to identify the vein and apply a tourniquet to find a vein usually in the arm. And fifth step is to clean the area with alcohol in circular motion. And sixth step is to draw the blood. Carefully insert the needle and collect the required amount of blood from your desired area. And the seventh step is stop bleeding. Remove the needle, apply pressure and place a bandage. And eighth step is to label the sample. And ninth step is transport the sample, and labeled bottles are placed in cooler bags to transport the sample in lab quickly to ensure good results. Incubation is crucial step in blood culturing because it creates ideal conditions for microorganisms to grow and multiply. The blood sample bottles are placed in incubator for 24 to 48 hours to encourage the growth of microorganisms. Many harmful microorganisms are present in small number may not be immediately visible in the sample. Incubation provides warmth around 37 degrees Celsius that encourage bacteria or fungi to grow and become detectable. Without incubation, microorganisms remain undetectable, so we provide incubation. After incubation of collected blood for blood culturing, the next step in the lab procedure are aimed at detecting, identifying, and analyzing any microorganism that have grown. The lab technician check the blood culture bottles for signs of microbial growth such as cloudiness, gas bubble, or any changes in the color. The growth indicates the presence of bacteria, fungi in the blood sample, which help in diagnosing infections like sepsis. In gram staining, a small amount of blood culture sample is taken and applied to microscopic slide. The sample is stained using a method called gram staining to differentiate between bacterial types. This helps the technician to quickly categorize the microorganism either in order positive to or see negative. Isolated colonies, we do subculturing. A sample from blood culture bottle is transferred to solid agar plate to encourage isolated colonies to grow. Isolated colonies allow for better identification of specific organism and help in further testing such as biochemical tests. After identifying the bacterial growth, the technician performs antibiotic susceptibility testing like disc diffusion or broth microdilation to see which antibiotic will kill the bacteria. And very last step is reporting the result. The technician compiles the result including the type of microorganism found and its antibiotic susceptibility profile and send the report to attending physician. This information is crucial for physician to precise the right treatment and manage the patient condition effectively. I hope you have enjoyed the video and thanks for watching.